The overall goal of this surgical implantation of electrodes is to record central and peripheral bioelectric signals simultaneously from a freely moving rat.
This method can help answer key questions in the neuroscience field, such as memory, cognition, emotion, and brain-body interactions. The main advantage of this technique is that the single recording device collects all the bioelectrical signals from the wide range of body areas.
We first had the idea for this method when we noticed that muscle and cardiac activities are both represented by electrical signals similar to brain extracellular signals. We therefore considered that all signals can be recorded by a single multi-channel recording device.
To begin, prepare a standard microdrive array for cortical LFP recordings. Leave at least 6 metal holes open on an electrode interface board for the signaling channels. Next, make 6 5cm lengths of BioFlex wire to carry the signals. Peel off about 5 mm of PFTE coating on both ends. Then, connect one wire to each metal hole in the board using a gold pin. Next, make 2 5cm lengths of enamel wire and solder them to the 2 ground reference channels on the board. To make the ECG electrodes, cut 2 16cm lengths of BioFlex wire and peel off the 0.5 mm